Welcome back. I hope you enjoyed lunch. Again, a quick reminder, please. Turn off your cell phones or turn them at least into silent mode, and as much as possible, try not to use them too much in this room. You can check your messages at our next break in a short while. So, we're going to move on to some more rare diseases this afternoon rather. So let's see, really from the family's perspective here, the challenges of having a child with a rare disease. I'm going to show you a little video that will set that up, and then we'll move on to our afternoon panel. And you might, if you cry at a Budweiser commercial, you might want to take your tissues out for this particular video. So if we can start that video, please. We've been talking about rare diseases today in mostly scientific terms. That is, after all, why we're here, to share, learn, collaborate, and hopefully move closer to cures. But let us not forget the real reason we're here, the patients and families impacted by these rare diseases. So here's a brief reminder of what it's like to live with a rare disease, so that we don't lose sight of the ultimate goal, alleviating their suffering.
We had these amazing, they're still amazing. We had these amazing children who are normal in every way, full of life, exuberant, driving us crazy, but we're loving every minute of it. And, then one night, it's almost like a thief broke into our house, and he started stealing pieces of our son. He'd come one night and take a few words, and at first we didn't notice. But then as it added up, he took more and more words. A few months later, he took Noah's ability to walk, and he started stumbling more. Within a few weeks, that ability was totally gone. The thief came back. A few weeks later, took his ability to eat. It's taken his ability to see, to talk to us. It's now, at this point, taken nearly everything.
Noah Van Houten was a normal, happy 3-year-old when he started having seizures, the beginning of a devastating downward spiral that led to a diagnosis of Batten disease, a rare genetic disorder that causes progressive brain degeneration. It is a terminal disease with no cure. 5 months after Noah was diagnosed, one of his younger twin sisters was also diagnosed with Batten.
It is a cruel, cruel disease. No parent should have to go through this. No child, more importantly, should have to go through this.
Olivia Birtwistle was a bright, vivacious 7-year-old when she suddenly went blind, often the first sign of Batten disease.
These kids, they're bright. They have dreams and want to do all these things, and then to see them robbed of it is one of the most devastating things. This is the disease. This is not what Olivia would choose to be like. And I think that's really what's so frustrating for these kids is they know what's happening, and they know what they used to know, and they can't do a darn thing about it.
We just need some help. We need some people to get interested in this, and we're determined to make it a difference in this disease.
A very different but no less devastating rare disease is often called butterfly disease. That's because the skin of these children is as delicate as the wings of a butterfly. But unlike butterflies, there is nothing beautiful about this disease. 14-year-old Jonathan Petra is a butterfly child. His mother recalls when she first heard the medical term for her son's disease.
Jonathan was starting to have little blisters on his hands. We didn't know what was causing it, and by the time we got the CHEO, he stopped breathing because he had blisters all of his in esophagus. They did a skin biopsy in which came back saying that Jonathan had EB, which is epidermolysis bullosa.
EB is a genetic connective tissue disorder that leaves children with skin so fragile that the slightest touch can cause blisters, tears, and open wounds that are easily infected. This is a brief glimpse of life for an EB family from a Barcroft Media profile of Jonathan and his mom.
The day-to-day challenges of EB is everything, really. To eating, to walking, to getting dressed. This condition is very rare. My skin in general, the wounds had gotten bigger, deeper, and now they're chronic wounds, which means that they never leave.
Wound care is an arduous, hours-long daily undertaking for all EB children.
The bath is horrendous. It takes a very long, 3.5 hours, like approximately, sometimes 4, sometimes 3, depends. The pain has gotten dramatically worse.
He soaks in the bathtub with salt, pool salt, and Javex. Javex being a disinfectant in the pool salt. Once he were done removing all of his dressings, he soaks in the tub for a few minutes just to have a bit of soaking time. We get him out of the tub, we walk to the room, and it's doing the dressings over again. So we start from the feet up so that eventually once I have covered his bottom, he can actually sit down because it's very hard for him to stand for a long time. He tries to imagine what a day without pain would be. I can't even picture myself in being that amount of pain every day of my life.
Jonathan knows EB will likely take his life by his mid-20s. Yet despite, or perhaps because of his EB, Jonathan is wise beyond his years.
I'm already past half my life. So when you think about that, you know, it gives you a new perspective. That's why you should live life to the fullest. That's why you should not, always, think about what you don't have. Think about what you do have. Think of all the happiness around you. Think about all the love, not all the negative stuff. Think about the positive stuff, and you'll be a very happy person.
Okay, good. Welcome back from lunch, and thank you for joining the afternoon session. I think the video that we just saw did a really good job of capturing what the children and their families go through. So now we're going to look and turn our directions towards the hunt for new disorders or new cures for many of these rare diseases. As Dr. Smith noted earlier this morning, the fields of cell therapy and gene therapy are intersecting. And this is particularly true in regards to research on the rare genetic disorders. So today we've asked a number of researchers and clinicians to come together and pull together a team of the world's best scientists and researchers to really talk about these two approaches. We've sort of separated them, not that they don't get along, but we're going to see what the intersection is, and maybe by the end we'll bring everybody together into one group. So we're really excited about what we're going to be talking about today. And looking for better ways to collaborate and speed the delivery of the cures. If I had to read the biographies of all the scientists and clinicians who are joining us, we would take most of the whole hour that we have for this. So we'll just keep our introduction very short to the names and the institutions. Together they have over 500 scientific papers and some of the best in the world that have been published in these different areas. And their complete biographies are found in the conference program. So we'll continue in that line and give them the time rather than me spending time introducing them. So representing the gene therapy approach, we have Dr. Sven Kili, who works in ADA, adenosine deaminase deficiency, severe combined immunodeficiency, as GlaxoSmithKline's Vice President and Head of Gene Therapy Development. Joining him is Dr. David Pearce, President of the Sanford Children's Health Research Center and one of the world's leading figures on Batten disease. And finally, Dr. Bill Hauswirth, the Rybaczki-Bullard Professor of Ophthalmology at the University of Florida College of Medicine, who's done unique work with ocular disorders. So on the other side, representing the stem cell group, we have three other luminaries. Dr. Jill Weimer, who's working on ataxia-telangiectasia, or AT as it's commonly referred to, and is a director and scientist of the Children's Health Research Center in South Dakota. She'll be joined by Dr. Jacob Tolar, professor and director of the University of Minnesota's Stem Cell Institute, who's doing promising work in epidermolysis bullosa. And this is better known as butterfly disease. At far left is Dr. Joanne Kurtzberg, who you heard from earlier. She's the Chief Scientific Officer and Chief Medical Officer from Duke University's Medical Center, and a leading figure in hereditary metabolic disorders. So we'll get started with the individual presentations. I think if we could get this.
So thanks very much, Stephen, and thank you very much for the kind of an invitation to come and speak. I'm going to talk today a little bit about a gene therapy for ADA-SCID that we've been developing, but also just give you a little bit of an overview of some of the other activities that are ongoing. So first, I'd like you to imagine you are the proud parent of this beautiful little baby child. Child comes out, everything is fantastic, perfectly formed, 10 toes, 10 fingers, absolutely amazing. And then a couple of weeks go along and the child gets sick. It's not a big deal, all children get sick. So you go off to your general practitioner, get some antibiotics, child gets better, fantastic. Then they get sick again. So you go through the whole thing again. Then they get sick again. And this time it gets more and more serious until eventually you find yourself in the intensive care unit with your child strapped up to a lot of monitors, tubes, all sorts of things, trying to figure out what is going on. This is very often what happens to children with ADA-SCID. So adenosine deaminase, severe combined immunodeficiency syndrome. And this is one of the very rare and really ultra-orphan diseases that affects a very small number of children, about 14 in Europe per year and about 15 in the US. So what are your choices if you manage to get your child diagnosed soon enough, if you can have the child diagnosed before the age of 2 when they often perish? Well, you could look at allogeneic hematopoietic stem cell transplants or bone marrow transplants also. And if you have a matched sibling donor, you're actually in really good luck because the results are phenomenally good, about 90%. That's really great. But as you start going down the scale and having unmatched donors and things like that, your results start dropping off very dramatically. So you could also go for enzyme replacement therapy. The problem with that is enzyme replacement therapy is not approved in many countries. And so that may limit your choices. But more recently, your alternative might be to look at gene therapy. And this is something that we've been working on with our partners at the Telethon Institute of Gene Therapy in Milan. So what is autologous gene therapy? For those of you that are not familiar, the patient comes in and they have collection of the hematopoietic stem cells from the bone marrow. These cells are then purified to get the CD34 cells. These cells are then transduced. And what they have done, the transduction happens using a vector, which is a virus. So for this program, we use a gamma retrovirus. For the other programs I'll discuss very briefly, we use a lentivirus. And this virus really serves as a transport to get that new genetic material in place. That's tested, and whilst it's tested, the patient is prepared with a gentle chemotherapy to get them ready, and then those cells are reinfused and the patient is followed up. So I'm very excited to sit here today and tell you that on the 1st of April of this year, our product for ADA-SCID was given a positive opinion by the European Medicines Association. And so all going well, we should have a license within a month or 2. And so this is really exciting news because this is the first life-saving gene therapy for children anywhere in the world. The data, very briefly, is 100% survival at a median of 7 years. It builds on our capability, and you can see in the slides, we have a very dramatically increased T cell count, which is really what these patients are short of, and also as a result of that, very reduced infection rates. We've had 100% survival, and of that, we're incredibly grateful. In addition to the ADA-SCID program, we also have a number of others. We have one in Wiskott-Aldridge and one in metachromatic leukodystrophy. We also have studies ongoing in beta thalassemia, and then 3 more preclinical studies on the go. So there's an enormous amount of work all in these rare diseases. And just to finish off, give you an overview of one of the others in metachromatic leukodystrophy. This is a particularly devastating disease, which I won't go into in any detail because of time limitations. But the clinical study that we have includes late infantile and early juvenile patients. And the slide that you can see and the graph that you can see is looking at late infantile patients. To date, we've treated 9 of them. Unfortunately, only 7 are shown on this because the full 9 haven't been published yet, and so I'm not allowed to present it. But 6 of those 7 showed normal function. 2 showed subnormal function, and 1 showed a low functional status. And so what you can see, if you have a look at that diagram, the gray shaded area is the normal gross motor function development of children. That's what you would expect to see. And all of the colored dots are the patients that have been treated. So that's the 7 of the 9 that have been treated, you can see. So 2 haven't done so well. As I said, the 1 that's low functional is below those. But what's particularly interesting is all of those orange dots lying along the bottom. Those are the siblings of the patients who've been treated, because MLD, very often you can't diagnose until you've had a sibling that's already been affected. And so those are the ones that are at the bottom. You can see their level of functioning. And so at this stage, all of the patients in the trial have survived past the death of their siblings. So we're really, really excited about this. There has been 1 fatality, but this was unrelated to the treatment. So you'll be seeing a publication coming out on that in a few months. So as I said, we're really, really excited, and we're very happy. And I just want to leave you with a bit of good news. This is Sebastian, who had ADA-SCID, and he was treated. He was the very first patient ever to be treated. He's now 15 years post-treatment, and he's absolutely loving life. And so we're really, really grateful for our collaboration with the Telethon Institute. Thank you.
Thank you, Sven. And we're going to hold the questions for the 3 gene therapy researchers until they're finished, all 3 presentations, because we may get some overlap. So we'll see what everyone has to say, and then we'll switch over to the stem cell therapy. So our next speaker will be David Pearce. We'll be talking about Batten disease.
Okay, thank you, Steve, and thank you to Robin and the organizers for having me present today. So I'm going to talk a little bit in general about rare diseases, just really to punctuate some of the points that Dr. Groft did in his overview, because I think it's important that we really remember some of these numbers. But I'm also doing that to really let you know that we keep hearing about the problems that we're beset with in rare diseases. So I'm going to just generally tell you some of the solutions that we're coming up with for rare diseases on the whole before I move into the specific clinical trial that I discussed. So we know that most rare diseases are really simply treated off-label. They're treated symptomatically. And Steve also pointed out, well, because of the rarity of these diseases, we need to put these patients in a registry. Well, I look at that as combining both of those things. What we've established at Sanford is the Sanford Coordination of Rare Diseases registry, where we actually build the registries for patient groups, because the patient groups don't have the resources to do this. And the other important point to do that is not just for each individual disease to have that rare registry so you can track and then actually instigate a clinical trial for that one disease. I just said it. We treat most of these diseases symptomatically. If I could tell you all of the different rare diseases that use different anticonvulsants without us truly effectively monitoring how these seizures are being treated, let's put all of that in one rare disease database, and then we can monitor that. So it's more of a global way of looking at things. Suddenly, from one rare disease, you can have maybe 1,000 rare diseases with the clinical information that you can track in one foul swoop. So we're moving forwards with that. And I talk about children because it was emphasized. 2/3 of the individuals, of the 30 million Americans, and I'm afraid I don't think I know the global number, but that's 20 million children in the United States who are afflicted with a rare disease. Cumulatively, that's a significant number. So just on the right-hand side of the slide there, we have a children's research group that Jill and I are a part of, and a number of other investigators where we actually, are really going from bench to bedside with respect to a number of therapeutic strategies right now for rare diseases. And currently in the Rare Disease Registry, we have 300 rare disease registries online. So switching gears, I'm going to talk specifically about Batten disease now. So like Sven, I'm just going to put this into the context for you right now, but not in terms of a picture of a child. I want you all just to take a moment and think about one of your children or one of your grandchildren, or just a child that you know, age 2 or age 5. The personalities, what they're doing, all of the different fun things that they do. Different forms of Batten disease can start to manifest at age 2 or 5. Just picture that child that you know right now. They'll start to lose their vision. After losing their vision, they'll start to decline mentally in some ways. They'll forget your name. They'll forget their own name. They'll have seizures. they'll lose the ability to speak completely, they'll end up in a vegetative state. These diseases are universally fatal. That's unacceptable to me, absolutely unacceptable. So what we're doing with these different diseases is really coming up with different ways to approach this problem, because the genetics are, it says here, there's 14 different forms. That means that you have a child that you are, unfortunately, out of the 3.2 billion letters that make you up, probably one of those letters was the same in you that was which was wrong, it was a mutation, and your partner was mutated as well. 3.2 billion and one change can cause these diseases. So we could say gene therapy, and I'm going to talk about gene therapy, but you need to throw the kitchen sink at this. You need to throw different ways. So natural therapies, for example, let's just not forget aspirin was once known as tree bark. We're isolating these compounds. So we can try to modulate that sort of organelle. This is something, the geeky part of this is a part of the cell that we know doesn't work. It's called the lysosome. We can look at anti-inflammatory. The brain is inflamed in all of these diseases. We can try and modulate that. We can do drug screens. We can try and go to the source of the mutation and modulate the DNA or RNA. And we can do gene therapy, which I'll just pass on because I'm going to talk a little bit more about that. Or you can manufacture the actual protein and inject it. And then Jill will be talking a little bit about stem cell therapy approaches. So, no animation, that's going to make it a little bit more difficult. Okay, so what we have here is a very busy slide. If I walk you through the top left-hand side here is this. Gene therapy for this late infantile variant of Batten disease, CLN6. It's never been done before. So the first thing you have to do is just take the human gene and just force it into the brain of a mouse to see what happens. The dirty secret of gene therapies is you're putting a lot more into the cells than actually normally exists. And sometimes that can be a bad thing. So you want to make sure it's safe. So the first slide just says you get the gene in there, it's not killing the cells, it's good. The second panel really just shows the orange bars is how much of the gene corrective DNA we're putting into the cells, and the whitish sort of bar on my screen here is just the normal level. So we're putting a ton more in there, and that's okay. Moving across to the blue and the green, that's a cross-section of a mouse brain. Green is the way we track. Did we get the virus in there? This virus is a stripped-down, safe way of just delivering the DNA. It's kind of taking a Ferrari and turning it into a minivan. So you can put the kids in there. You're taking all the bad stuff out, the speeding and stuff that you don't want, and making it safe. So it's across the whole brain, as you see. The squiggly line thing on the far right at the top there is where it says WT is wild type. There's a single line going down there with all those little bobbly bits down the side. They're all the dendrites that are firing and connecting and making your brain work right now. The middle one is the disease in the mouse, and you see there's not many bobbly bits, so it's not really firing. And with the gene therapy, you can see there's a lot more. Down the bottom left is, because I know I'm sort of running on time here, is across the bottom there is just our way of showing in red that we've managed to get the virus to express the gene into multiple different cell types. There's no point putting the virus in there if it just floats around in the brain and doesn't actually get into the cells, into the neurons that are doing that firing. So this is effectively done. The panel in the middle of the bottom there is that stuff, it says a wild type there, that black box, that's what a normal brain should look like with this particular test. And the glowing green is in the disease, and what we call autofluorescent storage material. There's just garbage growing and glowing, accumulating in the cells of the brain of the disease. And then when you put the virus in there on the top right, it goes back to being a black screen, which means you've reversed the process, and you've cured that biochemical defect. The other panel is just below it, just other tests to do it. I just draw your attention to the one on the bottom with the blue. Again, in the wild type, there is nothing there. In the disease, you have these red dots, which is the accumulation, another way of looking at the pathology, and the virus has cured that. The money slide here really is that if you look at the histogram or the bar chart on the side there, is that the whitish pale is really what you would see in just a normal mouse. And then the blue is the disease. That's how much disease is present. Put the virus in, and the orange returns you to normal. So pathologically, what we've shown is that you can reverse the storage material. And of course, we did a lot of other things in terms of safety and toxicity. And this preclinical data was presented by Jill Weimer and her group to the FDA. And I'm proud to say that last week we just treated the second child with this gene therapeutic at Nationwide Children's Hospital. in partnership with them. We can't identify those two individuals just yet, but what I can tell you, the first patient that was treated 5 weeks ago, we did get a message from the father saying she wasn't walking, she just took some steps. So think of that child that you had in your mind and what the capabilities are, because that's what this is all about.
Great. Thank you, David. Our third speaker will be Dr. William Hauswirth. So, Bill, go ahead.
Thank you. I'm going to speak about a whole field of gene therapy that involves developing cures for blindness. And we're going to talk about single gene blinding diseases, that is, diseases that are caused by mutations in one gene. And I'll give you a slide later to show you how many of those single gene blinding diseases they are. In general, then, this is retinal gene therapy. Retina is an organ that lines the back of the eye. And I think I need to start with an anatomy of the eye, because not only because it's beautiful, this is a human retina, the central 5 or 6% of the retina. And what you see here is something counterintuitive. Light is coming in from the top. That is where our world is coming from as far as vision is concerned. But the cells that receive the light, the rods and cones, are at the bottom of the retina. So in fact, light needs to pass through other retinal neurons first before it reaches the rods and cones. But in the human retina and in all primate retinas, we have a very special structure in the middle of the retina called the fovea, where those inner cells, where those cells that see the light first are moved to the side during late development. So in fact, that gives you a hint as to where the most important vision function is. It's in the fovea, because that's where evolution has moved the cells aside, so there's not as much distortion of the image when it reaches the rods and cones. Now, in the fovea, and only in primates, in the center of the fovea, there's less than 1/2% of the fovea, there are only cones receiving that light. This is pure cone retina. Outside this cone-only region, there are mostly rods. Rods mediate night vision, low light vision. Cones mediate daylight vision, high acuity vision, color vision, most of our motion sensitivity. So cones are really the cell that defines the status of your vision. Most of human vision is due ultimately to loss of cones, these foveal cones. Enough on the anatomy. The anatomy is important because our vectors are recombinant viruses that contain none of the original genes. They contain the one gene that we need for the given disease. But we can manipulate what cells express that gene because we know what cells have a deficit in each of the diseases. So cone diseases, you want to target cones, as I show you there. For rod diseases, you'd like to target not the central cones because you want to target where the gene is missing. Those are rod diseases out to the periphery. In the ganglion cells, which have moved to the side in the central, you have a number of other diseases which are primarily ganglion cell diseases, and I include glaucoma and diabetes here. They are not rare diseases, but just to give you a context of where common diseases of blinding diseases are. But mitochondrial diseases are rare diseases. And finally, a single monolayer of cells called the RPE cells. And for comparison, AMD is not a single gene disease, nor is it uncommon at all. But just for reference, I want to show you where they are and what cell types they affect. So let's take a look at what diseases are currently in gene therapy clinical trials. At the top, you see a ganglion cell disease. This is a mitochondrial disease, Leber hereditary optic neuropathy, LON ND4. For rods in the middle, there's an autosomal dominant retinitis pigmentosa trial, a Stargardt disease trial, and Usher 1A trial. By the way, these are all rare diseases by anybody's definition. And the bottom, you see in the RPE, that single monolayer of red, you see RP65, MERTK, and choroideremia, and again, AMD is down there, but we're treating the symptoms, not a single gene, because there's not a single gene that causes it. What I want to discuss in just slight detail is the first gene therapy clinical trial for blindness, and that was for the RP65 gene. I should point out that in this slide, the childhood forms of blindness are the LHON at the top, the Usher 1A, which is a syndrome, which is blindness and deafness at birth, so a terrible condition, and the RP65. So let's look at RP65 in just some detail on the next slide. And I'm doing this because this was our trial. It is the longest running trial for gene therapy for blindness. We treated 15 patients. 12 of them benefited by as much as 60,000-fold. That meant that in the region we treated, they could see 60,000-fold dimmer light than they could before treatment. I'll give you a couple of examples of what the patient said. One patient said, I don't know if you have fireflies in Europe, but we do in the United States, especially in the Middle West. She came back excited 1 month later and said, I can see fireflies for the first time in my life. That doesn't sound like much, but it's for her, it was all the world. Another patient, an 8-year-old, came back in about 3 months later and said, I have a flashlight in my vision. Cool. And it turned out we asked him to draw on a map where he saw his flashlight, and that's exactly where we had put the virus. The vector is not put in over the entire retina. It's put in a defined 15% or so of the retina. Importantly, 1 year after treatment, all of those 12 patients showed stable improvement. We have then followed them for up to 6.5 years, and the vision benefit remains from 100-fold to 2,000-fold. But as you can see, there's been a slow decline in their vision from when they first when we first did our first follow-up study. And I believe this is an issue that was brought up before. In an early phase study like this, we don't know the exact dose, and we don't know the stage of disease we should be treating. And so I believe the slow decline is due to insufficient vector and treatment too late in the disease, and so this will be corrected in the future. What's left to do? Well, the genes causing human blindness have been analyzed for about 20 years now, and you can see over time there's been almost an exponential increase in those genes. Identified genes today are over 250. That is, there are 250 genetic causes, different genetic causes of human disease. So how does that relate to what is being done today? In animal models, which we do have quite a few animal models, we have cured about 35 different animal models of blinding diseases, about 25 of them in my lab over the last 20 years. More importantly, what's in clinical trials? There's about 8 in clinical trials today, so you can see immediately that as far as rare diseases are concerned, there's a huge therapeutic opportunity moving forward. And finally, what's coming up in the near future, in the next year or two? There are 2 rod diseases that will be in clinical trials soon. X-linked retinitis pigmentosa, which is a study sponsored by the gene therapy company AGTC, and that's a childhood form of blindness, as well as LCA1, which is an early childhood form of blindness. It's a partnership with University of Florida and Genzyme Corporation. And then cone-specific diseases, there are 3 that are just started clinical trials, 2 forms of achromatopsia, A3 and B3, and 1 form of blue cone monochromacy. These were supported by the Foundation Fighting Blindness early on, or by the BCM Families Foundation, and are now are being supported again by AGTC. So that's a quick overview of basically the whole field of retinal gene therapy.
Okay, good. Thank you, Bill. We have a few minutes for questions. So first, Sven, you're anticipating approval.
We're very hopeful.
You got it. Okay. So where can the patients go to be treated?
So currently, the way the product was set up and manufactured, it's still delivered in essentially what's called the fresh form. So patients are currently still required to travel to Milan to be treated, where they need to stay for a number of weeks for the full treatment regime. For the ADA-SCID product and for all of the others, we're in the process of developing a cryopreserved product so that patients will be able to be treated in their home countries at their own specialist centers. But that's a little way away still.
Great. Yeah, because for a lot of patients, travel is a significant problem. So, yeah.
It is, but what we found even for the clinical trials, we've had patients from a far afield as the US, the Gulf states, everything like that. These, they're willing to travel, and the company, has supported them. So if it means getting a one-step treatment that potentially opens them up to never needing treatment again, very often they're willing to do it.
I have one question, and we'll extend the first question. So first, Bill, how do you decide between gene and cell therapy?
the decision between gene and cell therapy is often made for us depending on the stage of the disease. In many of the diseases I work on and in many of the other CNS diseases, there is a degenerative process where cells are lost. So there would be a stage, especially in the blinding diseases, where there are no affected cells left. In that case, gene therapy will not work because you don't have a cell to deliver the gene to. Therefore, you must use cell therapy or some alternative. And in the eye, there are ways being tested, not in patients yet, to make other cells light sensitive other than rods and cones, which are lost in these patients, and to substitute light sensitivity in another retinal cell that hasn't been lost. And the initial experiments in animals show that the brain can make use of that information, even if the cell that's receiving the light is not a rod or a cone. It's very interesting, and it has a ways to go.
Okay. And David, taking it one step further, so we've talked about cellular therapy, gene therapy, and then enzyme replacement therapy. So how do we decide?
That's a great question. I think Bill nailed it partly in terms of the retinal diseases. In Batten disease, the brain is literally falling apart, and it's a very progressive, slow disease by some counts. So obviously, for gene therapy to be effective, you want to be able to intervene as early as possible because you need to be able to have cells there that you can actually get the virus into. I mean, ultimately, cell therapy, and in terms of some of the stem cell work, is if you can actually replace the cells that are being lost, that's really the ultimate. I think my feeling is for Batten disease in particular is it will be combinational therapies. The first clinical trial that we completed for the juvenile form was actually a small molecule therapy. With the data coming in from that right now, we expect that we'll probably combine that with potentially with a gene therapeutic approach similar to the one I just described. Accessibility, if you think about trying to penetrate the body, the brain, the muscle, the liver, all the parts of the body with different therapeutics, you can't just have a single fix. So there's really multiple approaches, which gets to that one slide I showed, that you want to develop each one, and as our knowledge improves, that we can apply multiple therapies. And the FDA is very open if you work with them on this type of approach.
One last question for Sven. What's the motivation for Big Pharma to work in these very rare disorders?
Yeah, I think that's it's a great question because Big Pharma typically, look at a company like GSK, where we're now able to treat maybe 20 patients per year in an ultra-orphan disease. It's not exactly something that's going to turn the needle from a financial perspective. But I think really what it allows us as a big company to do is we've in the last 5 years really focused on rare diseases for a number of reasons that people were talking about this morning. Is rare diseases really help us to understand the biology and understand the pathophysiology of a number of more common diseases, and that in association with really understanding that the cellular therapies and using a patient's cells and a patient's genes to be able to hopefully permanently correct disease is really the fourth pillar of being able to treat patients. So we've got small molecules, we've got large molecules, and now cell and gene therapy. And so this is really an area that we're very focused on. And we look at these first initial indications as really the way for manufacturing to really scale up and to understand how do you manufacture these things. From the clinical development teams that I work with is really to understand how do you design clinical trials that are appropriate for these types of diseases. How do we understand the pathophysiology? How do we understand the translation and moving forward? And I think once we start really understanding that, it allows us to move forward into more common indications and really make it more of a continuum. So I think this is very much a long-term investment in understanding the need to really address these rare and ultra-rare diseases because there are so many patients out there deserving.
I'm going to punctuate with one very brief statement there, Steve, so my apologies. I think, I said this to some people earlier, you heard about specific point mutations and DNA changes this morning in cancer. Rare diseases, we've been studying this for the last 30 years. It's personalized medicine, so that's the expertise there. All of the other diseases that you heard about this morning ultimately are rare diseases, and that's really something that we need to consider in terms of the expertise and the application of understanding these diseases. So you will learn a lot. You heard a wonderful story from the young lady this morning about getting the samples, finding that mutation. It's the same in rare diseases, and we will learn about more complex diseases.
Great. Thank you. So we're going to switch now to the stem cell therapy, and Jill Weimer will be discussing AT.
Excellent. So the previous questions kind of set the stage perfectly for my presentation. You heard from my colleague David Pearce that at Sanford we work on a number of rare diseases, but we know that there's not going to be one cure, one pill that cures those diseases like you see with cancer. And so we're throwing the kitchen sink at this. So this is a good transition to cellular-based therapies as another form of treatment. And I don't know why my slides aren't coming up. I have to make them come up. So we're switching to cellular therapy, so I just want to, for this audience, I don't need to tell you that anything really about stem cells, and we had some great overviews this morning, but I just want to draw your attention to what we're doing at Sanford. So in the lower left-hand side of the screen, you see the two different types of stem cells, and really our work at Sanford is focused on adult-derived stem cells and what we can do with those, ranging from induced pluripotent stem cells to mesenchymal stem cells from fat, from cords, and sort of where that goes in the future. You will hear from two of our colleagues tomorrow talking about some of the clinical work that we're doing using adult-derived stem cells in clinical practice currently. And so I'll move forward just to introduce you. So one of the things that was highlighted in a couple of the previous speakers that we suffer in the rare disease field is that in some instances we have great animal models that we can use to screen those diseases. With the CLN6 therapy, we had a very good mouse model that we could use for that, but for even many of these diseases, we want to do either stem cell treatments or screening in animal models, and we don't have great animal models. And so what we've been doing in the innovative practices at Sanford that I want to talk to you about today are developing innovative animal models that we can use for screening adult-derived stem cells. And so the stem cells that we work with, we heard earlier from Joanna about these. So these are mesenchymal-derived stem cells. They can come from a number of sources. So on the left-hand side, you can see these can come from bone marrow, adipose tissue, amniotic fluid, umbilical cords. But these cells are restricted in their fate. They can only differentiate into certain types of cells. And the diseases that my group particularly works on in many of the labs at Sanford are rare neurological disorders. So these cells do not have the capacity to become new neurons, but they do provide a lot of potential benefits in the treatment of neurological disorders. So in the center schematic, you can see some of these. So many neurological disorders have a common theme that you have rampant neuroinflammation. So if we can modulate the immune system using mesenchymal stem cells, if we can tighten the control on activation of microglia, if we kind of move around the horn on this slide, mesenchymal stem cells can also stimulate endogenous neuronal stem cells to proliferate in the brain. They can allow for the secretion of trophic factors that provide support to these neurons. They can allow for synaptogenesis and strengthening of synaptic connections. They can allow for remyelination, and so promote remyelination in diseases like ALS. They can promote formation of new vasculature, which is something that's you see in many rare pediatric disorders. And they can also control or increase the activation of astrocytes, those glia in your brain that help to clean up a lot of this debris. And so, as I mentioned, what we've been working on is developing innovative animal models. And so many times in mouse models, which is the gold standard for basic biomedical research, we make mutations that we see in human patients, and they don't perfectly mimic what we see in the human patients in the mice. And so what we've been working on at Sanford is actually in collaboration with Exemplar Genetics to develop porcine models or pig models for these rare pediatric disorders that more closely mimic the human disease. So just quickly to run through the schematic, the way that we do this, in this case I'm showing you a human mutation in NF1, the gene that's mutated in neurofibromatosin type 1, is to introduce that into the wild type DNA of the pig, and using somatic transfer, we're able to reintroduce that into a pregnant donor recipient pig and create a herd of pigs that now have neurofibromatosis. So in collaboration with David Pearce's lab, we've been able to make 4 different pig models thus far. Those include CLN2, so the young man that you saw in the video, Noah, suffers from CLN2 Batten's disease. Olivia, the second child we saw, has CLN3 Batten's disease. In collaboration with the Children's Tumor Foundation, we've made a pig model for neurofibromatosis type 1. And the one that is most well developed and that we published on is a porcine model for ataxia telangiectasia. For those of you who are not familiar with this rare pediatric neurological disorder, these kids have atrophy within the cerebellum. That's the part of your brain that looks like cauliflower, sits at the back, controls balance and coordination, motor movement. And so these kids lose a specific class of cells within the cerebellum and have severe ataxia. And we see those same pathological and behavioral changes in our pig model. And so what we're doing now is isolating stem cells from the cords of the pigs and introducing them in a number of different ways. So the first, on the left-hand side, we introduce these cells by IV infusion. We look to see whether they elicit an immune activation in the periphery in a short-term study. And then using the same cord-derived stem cells, we look at long-term outcomes to see whether we can correct the behavioral and pathological output in this pig model. And then also knowing that the cells that we really need to protect and support are in the central nervous system, where it's using several routes to get the cells directly into the brain, including intranasal delivery as well as intrathecal delivery to deliver these into the brain, and using the same evaluation paradigm. So just in summary, we have these great pig models that are very unique, that we can study the use of not only cell-based therapies, but other drugs. And really what the ideal goal for these pig models is that we have something that's much more like a human, that we can use combinatorial treatments to screen in these before we go straight into humans. Thank you.
Thank you, Joe. Next, we'll hear from Jacob Toller speaking about epidermolysis bullosa. And I just mentioned to Jacob before the meeting, one of the leading advocates in the United States in the late 70s was a lady named Arlene Pessar, and her son had epidermolysis bullosa. And so she was one of the first groups of individuals and patient advocacy groups to really bring an emphasis to our Congress primarily, and then to the research community, the pharmaceutical industry, and FDA, the regulatory group. So if you can pass that on to the society, how grateful we are today. So thank you.
I'm delighted, that this is the case. So listening to everything here today and thinking about this a little bit, the first thing we need to do is to stop apologizing. This rare disease concept, that's an industrial market-driven term. There's nothing rare about the rare diseases. For once, they are the insight into very, if you will, common ones. It's a matter of perspective. All of those in the audience that take statins for cholesterol, that started with the rare disease. People who are looking for the risk for ovarian and breast cancer, that started with the rare disease, or so labeled. And my hope is that all that we do with so-called rare diseases will translate into something that's an enormous benefit for society. Second is that's not rare about rare diseases is the frequency. It may be up to, Stephen said, up 8,000 disorders. Okay, if you take them together, that's 10% of the population. That's an enormous burden. And just for comparison, all of us has used the word cancer. That's a very heterogeneous disease, right? It's not one disease as well. And thirdly, it's not rare, as Nicholas Jr. and Elena said this morning, it's not rare when it's your kid. It's 100% of your horizons. It's everything you are. It totally changes the dreams you had for your kid. So it's not rare. We are not doing anything rare. The second, I think that Cardinal Ravasi was right this morning, that there is a long-term connection between a humanitarian approach to life and science. The compassion is the bedrock of both of them. The difference, however, is that the reason we are here today and have anything to talk about started only about 400 years ago. That's about 15 generations of people. And some of these people who actually stand at the beginning of this enormous time, which is science, are Italians. Andreas Vesalius, Galileo Galilei, that started here, then Isaac Newton, others. That was the beginning of knowing that we cannot fix a human disease unless we understand it. That's why some of us have thrived to have the fluency across the basic science and clinical science, because there's really no difference. And we have to have, that. And the third is that, yes, this may seem to us that we are somehow special, but we are not. The patients are. The families are, no question, but we are not. We are only carrying forward the flame that started many, many generations again, and we just happen to be useful perhaps in some ways. Epidermolysis bullosa is a disorder that you saw the video of Jonathan Petrie, and you know what it can do. The molecular pathology of this disorder is on your left, the healthy skin is on the very left, and then the diseased one is on the middle panel. Basically, there is an attachment between the top layer of your skin and the deep layer of the skin. There's nothing firm about it. These are these loops called anchoring fibrils, and they are homotrimers, they are polymers of a chain-like protein called type 7 collagen. If the child is born without this, it's not like ADA. You don't wait 6 weeks, 6 months to know. Day 0, hour 0 of birth, will give you a child that you have expected with half of her skin gone, and that skin will never grow back. And that's just the beginning. Not only that they are in pain 24/7, not only that they get infected, they are bandaged from head to toe. They are, itching in an incredible amount of magnitude. This is totally destructive, to the family as well, because, when I come home, I go and take my boy for a play of soccer. When these parents come home, they take off the bandages of these kids, which is incredibly painful. So the calling, to do something, to push, outside of the normalcy of what we do in medicine, is enormous. The lesson for me, and we learn a lot from how to live our lives from our patients, the lesson for me was that the idea to come with the stem cells in this skin disorder, I treat kids with cancer for a living with bone marrow transplant, but it came from a mother, came from somebody totally outside of this. And the only credit we can take is that we didn't say, go home, be nice to them and stuff. But we took it to the lab, and as I said at the beginning, the science is at the beginning of this, right? We went to the mice, engineered them to have the same disease, proved that the hematopoietic cell therapy works for that, and then we turned it into the clinical trial. And we were able to show that you can correct the deficiency of the type 7 collagen with the hematopoietic cell transplant in 30, 40, probably 50 individuals. Jonathan Petrie that you saw on the video beforehand, is in the what we call workup before the bone marrow transplantation, one of the patients that we treat. The expectation, the responsibility that comes from doing this is that we are very honest about what we do, very translucent. So even though we do have a multitude of the experimental readouts, how to measure our success, we always look for more. So this is a diagram that is my vision of, how this should work. This is a combinatorial therapy. This is not just because we came up with something and the others came with something, that somehow one method is better than the other. The bottom of that is that you can use number of different cells. They can be from somebody else, allogeneic. They can be mosaic, they can be self-reverted, that, that are in the body, and they can be gene-corrected, gene-edited. And then we not only rewrite the genes into normal health, but we also can take the cells and reboot them into what's called induced pluripotent stem cells, and then use that as a platform, inexhaustible supply for any cell, hematopoietic, skin, and otherwise, to treat the children with the systemic or local therapies. So the history of medicine is important. We came from the drugs, that's about 100 years ago, then moved into the biologics, that's about 50 years ago, when we are supposed to be able to clone and fuse cells. But now I think that these two measures, as important as they are currently, has reached conceptual ceiling. And what we are at is the cells that can be programmed like a chip in your iPhone to go to the specific site in the body, like in the bone marrow transplant, and do something that has not been before done, like in the epidermolysis bullosa, which, to my knowledge, is the first example where you can take cells from one organ, that being bone marrow, and use them for the repair of a totally different organ, such as skin. So the angle that is actually important, to keep in all of this is that the responsibility that we all have, for children and adults with disorders that are not yet treated, comes back to the science of this, the lifeline of the medicine, and I am privileged and honored to be a small part of it.
Thank you. So the last speaker in this session will be Dr. Joanne Kurtzberg, who will talk about hereditary metabolic disorders. So Joanne.
Thank you. So I'm going to focus on a disease called Krabbe disease, which really is one of the most fast-moving forms of lysosomal storage disorders. And these diseases, which are also called leukodystrophies, often affect very young babies and progress rapidly, so you have very little time to effectively treat them. And I showed the graph this morning over on the left or something similar, where if you can intervene, and in the case of Krabbe disease, you're talking about in the first month of life, the red line shows 90% of babies will grow into adolescence and have variable neurologic outcomes. But if you wait until these kids are diagnosed after they've presented with symptoms, the green line, then only 20% survive, and they all have very profound neurologic deficits. And Krabbe is really a terrible disease because for mothers, their babies are so irritable they can't console them. And no matter how sick a child is, I think the worst thing is when you can't hold your baby and make them feel better. And so it really causes trauma to the family in addition to the child. Now, we've been transplanting children with cord blood transplant who have Krabbe disease for 20 years, and even when we transplant them in the first month of life, we've learned that some kids can be normal, like the little boy walking next to the stroller, but others, like the child in the stroller, even when they're transplanted early, still have major motor defects. They told us not to touch it.
You can always change.
Anyway, still have major motor defects. And so I became very motivated to help those kids like the kid in the stroller. And in the middle, you can see something called fiber tracking, which is a way to image nerve tracks. And these are the corticospinal tracks, the nerves that go from the brain to the legs, in two different 2-day-old babies with known Krabbe disease whose siblings had died of the disease. And the one over towards your left is normal, and the one on the right is abnormal, even at 2 days of age. So we know that if we do a bone marrow transplant on a child with that kind of tract, they're still not going to be able to walk, even if they survive. I showed this morning. When you get kids early, they can have very good outcomes, and that was the case in these two brothers who were transplanted before they developed symptoms. But when kids are later, we need a different therapy. I also showed you this morning on the left that we know that cells given in a standard transplant after chemotherapy in the blood do reach the brain. And so this is the brain of a child who unfortunately had symptomatic disease and died 10 months post-transplant, but was a girl and got a boy's cord blood. And you can see with the green dots that there are many male cells scattered throughout her brain. And we found this in the cortex, the cerebellum, the brainstem, pretty much anywhere you could look in the brain. So we know the cells go to the brain. And when we looked at what these cells were, we found that there's something called a microglial cell, and also some were oligodendrocytes. And so we went back to the lab, and we developed a cell therapy product, which is shown on the right, swimming around in the dish, which we call Duca-O-Cell version 1. And this cell therapy product is now in the clinic. And this cell can, in a mouse model where you can demyelinate something called the corpus callosum. So if you look at that box up in the upper left, that's a normal corpus callosum in the blue. And if you look on the bottom left, you can see that after we feed the mouse something called cuprazone, which is a copper chelator, the myelination goes away. And long story short, if we then give that mouse DUOC cells, we can make the myelination repair. So if you look over, it's hard to do this without a pointer, but if you go to the third one on the bottom in the middle, that's a mouse that got the cell rescue where the brain is remyelinated, and it's shown in different ways on the different panels. And so we studied this cell in many ways and showed that it replaces enzyme, it cleans up debris, it modulates inflammation and actually downregulates it. It causes oligodendrocytes, which are cells that make myelin, to proliferate and develop, and it promotes myelination. And with that information, we now have it in a clinical trial where we take a cord blood unit, which is middle panel on the right, and we use the big part for a regular blood transplant of a child with Krabbe after chemotherapy. And that little baby there is a newborn with known Krabbe because of a family history. And then we, 1 week later, take the small part of the cord blood unit on the bottom and in a special laboratory, grow the DUOC cells, which takes 3 weeks. And then 4 weeks from transplant, we give that baby a dose of those cells in the spinal fluid to augment the transplant and get cells to the brain quicker so that the part of the brain that's already damaged at birth gets rescued more rapidly. And the picture on the lower left is this same baby 5 months later, which was the point where his sister was vegetative and unable to do anything, and he is developing normally. He's now a year later and still doing well. And we've treated 7 patients with this approach and hope that this is going to more rapidly control disease. And we really hope that in someday we won't have to use this with a transplant, but that it can be a standalone therapy to help both children and adults with demyelinating diseases. So this is just one story along the way of, again, combining therapy, combining approaches, and step by step getting to a place where we can improve the outcome of these children's lives.
Thank you, Joy. So I think you have time for at least one question for each of the speakers. So we have additional time, we'll keep moving. But first, Jill, what are the challenges and barriers moving forward in the gene, particularly in the cell therapy fields of research?
Sure. So I think as scientists, our natural knee-jerk response would be money. I would actually argue that money is necessary but not sufficient to accelerate a treatment. So the gene therapy trial that David talked about, well, actually backing up, Stephen, you mentioned earlier that to go from start to finish, a normal timeline is about 5 to 10 years to do the basic science, the preclinical efficacy, and then to move into a clinical trial. Because of the chemistry and financial resources that were brought to the Salin-6 gene therapy project, we actually started that in August of last fall and treated our first patient the second week in March. That was because of generous financial support from Sanford, from the Gray Family Foundation, from Nationwide Children's, and from the NIH. So the financial backing was there, but it also was bringing together the right chemistry of scientists. And I think as scientists, we often try to go it alone. We're collaborative to an extent, but if we can bring the right people together to move these projects forward. The other thing is that, the NIH requires us to give them an annual progress report on how our work is going. The Gray Family Foundation for the gene therapy required us to give weekly updates on the progress that we had made. So when you have families saying, my children are dying, you need to move faster, it really does pull at your heartstrings and make you move much faster. So I would say it's a combination of things to get there.
How about the interactions with the patient advocacy groups?
Absolutely essential. So I think in the rare diseases, we're very fortunate that all of us are probably very closely tied to our rare diseases, our specific rare diseases. They are also out there lobbying for financial resources. So Tracy Van Houten, who they highlighted in the first reel, Noah's father, Tracy actually goes to the U.S., to the federal government, to the NIH every year to advocate for money for rare disease research. And so they're absolutely essential players in this process as well.
Great. And Jacob, you had some really, really nice improvement there, so thank you, it's really good. So is that improvement progressive, will continue, is it permanent, or are there limitations that you'll be seeing?
It's an excellent question because we didn't know this at the beginning. People need to realize, and I think majority of you do, when we open a new clinical trial, that's exactly what it is. It is an experiment of sorts. We don't know where this is going, and we design it as scientifically sound and as morally and ethically sound as possible, but we don't know the outcome. That's the whole point. So we didn't know at the beginning whether these will be permanent or progressive, these benefits, but that was the idea, that you use the stem cells at the beginning, as we do in bone marrow transplant, so that you have one-time therapy, and the stem cells will take over the production of these all-important cells that are made in the bone marrow and can migrate to the injured skin and epithelia and produce the type 7 collagen. We've transplanted the first child in almost 10 years ago, and I can tell you that the majority of these individuals have progressive, lasting benefits.
Great, thank you. And Joanne, moving forward, what messages should we be putting out there to really generate that interest and support in all sectors? I mean, the private sector, public sector. Any suggestions for different messages that we should convey and work on?
Well, first of all, children are important. And I don't think we take care of our children well enough, at least in the U.S. and our society. I think rare diseases in children deserve the attention, even though these are not taxpayers and these are rare instances and rare individuals. The things you learn and the people you help will go very far. Second of all, I think we need multimodal therapy. I don't think one approach is necessarily going to fix all of these diseases. And, you know, I grew up in the time when childhood cancer just became curable, and that was with multi-agent chemotherapy, not one drug. The first drug that worked, 3 months later, those children relapsed. But when you combine drugs, you could cure now 90% of kids. And I think in these rare disorders, we have to take the same, maybe broader approach. Maybe it's gene therapy and cell therapy and others, but I think it needs multimodal therapy. And third, I'll just say that the traditional granting situation, which funds most of the research in the U.S. at least, is not really focused on funding this kind of research. And the early clinical trials are not funded through government funding, and the foundations and the disease advocacy groups have really been the source of funding to make progress. And fourth and last, I would say that we need new regulations. We need faster pathways to approval. We need a new design. You can't do a phase 3 300-patient trial when there's only 20 patients with the disease. And so really a lot of those sort of pathways have to be redesigned and reevaluated.
I think we can keep our eye on what's going on in our Congress with 21st Century Cures and the new initiatives. I know we all are hopeful that there will be an increased budget allocations for this research, in particular an emphasis on rare disorders. So I think it's important to mention for all these disorders that to convey the feeling that if you're a patient with a rare disease, a family with a rare disease, to realize that you're not alone, that there are people throughout the world who we want to connect you with. And I think all of the panelists today, you've heard from them, and they're really unbelievably sincere individuals who've dedicated their lives to these diseases, as have most of you, and continue to do so. And so I think drawing on the strength of all of us, another saying that we have is no disease is too rare to deserve treatment. So we really want treatments for all of the rare disorders. And I know so many people and patients and families that I've spoken with over the years, we keep looking for gene therapy, for cell therapy, to give us the answers. And you've heard of some of the tremendous progress, and I think we'll be seeing those applications. And so thank you very much for your presentation today, and thank you for your continued commitment. These are unbelievably difficult problems, and thank you to all the members of the audience, as you've heard this morning from others, for your commitments and support for what's going on. So thank you very much.
Thank you.